Hello class, this is a quick lecture on the types of photosynthesis. Your outline is as follows. We're going to learn three specific types, one of which we've already learned about: C3 photosynthesis, C4 photosynthesis, and CAM photosynthesis. Now C3 photosynthesis is the classic type of photosynthesis, which is the one we've already learned about. Carbon dioxide simply diffuses into the leaf mesophyll, where it's immediately used by the Kelvin cycle in the chloroplast stroma. When we see the figure on your screen here, it shows a leaf in which we're zooming in and looking at the stomata on the underside of the leaf where gas exchange is occurring. And these stomata can open and close. Some of the factors that can lead to the closing of a single stoma is drought and the hormone abscisic acid. And this abscisic acid is produced in the leaves when plants are under stress. Now, the first stage of the Kelvin cycle is fixation. And this stage involves the combining of RUBP, which is a 5-carbon sugar, to carbon dioxide with the enzyme Rubisco. Now, C3 photosynthesis is called C3 because it results in the formation of a 3-carbon compound that contains 3 carbon atoms. And this compound is called phosphoglyceric acid, which is abbreviated PGA. Recall that all of this occurs in the stroma when we're doing the Kelvin cycle during classic photosynthesis. Normally, carbon dioxide diffuses into the leaf from the atmosphere. However, stomata on a plant will usually close down on a hot, dry day where there's lots of sun. But if you close your stomata, then you can't get carbon dioxide. And if you don't have carbon dioxide, then you can't build sugars. Photorespiration is an enigma of photosynthesis. We don't really understand why it happens or why it was selected to be in plants over evolutionary time, because it's a bad thing, and yet we see it in lots and lots of different plants. Now, photorespiration is a bad thing because it provides no energy for the plant, but causes some of the carbon dioxide that was destined for photosynthesis to be released from the plant before it can be used in forming any sugars. Recall that RUBP, which is that 5-carbon sugar, is essential as part of the Kelvin cycle because it's what carbon dioxide gets fixed to. But during photorespiration, oxygen gets added to RUBP instead by that enzyme Rubisco. Therefore, because there's no carbon that's found in an oxygen, then we're going to have no sugar that gets produced. Because without carbon, we can't build our sugar. So any carbon dioxide that might have been in the plant cells to begin with is essentially wasted. So to overcome photorespiration, different plant groups have evolved some different photosynthetic pathways. First, let's look at C4 photosynthesis. C4 is an adaptation to both heat and drought in the environment, and it is best seen in the leaves of plants that exhibit what we call Cran's anatomy. Now, this anatomy involves some cells around the vascular tissue, and we call this the bundle sheath. These bundle sheath cells have chloroplasts that are very large and are mostly composed of stroma. And these bundle sheaths, as I mentioned, are just found around the vascular tissues, usually found in the center of the leaf mesophyll. So what types of plants have this type of anatomy? Well, we see things like tropical grasses, such as sugarcane and ancestral corn, exhibiting this anatomy. And because of it, they don't do photorespiration, even at high temperatures. So how does this work, you might ask? Well, by concentrating the carbon dioxide at the bundle sheath, it ensures a relatively low oxygen concentration, thus minimizing photorespiration. So this diagram shows us how C4 photosynthesis works. First up, the main difference of C4 from C3 is that during C4, carbon dioxide is fixed to PEP, not RUBP, into 4-carbon organic acids by PEP carboxylase, which is a special enzyme that is different from Rubisco, which is what we see in the C3 photosynthetic pathway. And all of this is occurring in the mesophyll, so it's not occurring in the stroma like we saw during C3. Now the important thing here is that PEP carboxylase is an enzyme that is not sensitive to oxygen, and because of this, it minimizes photorespiration. Now, carbon dioxide, which is stored in these 4-carbon compounds, can be transported to the bundle sheath where it can enter the normal Kelvin cycle. Now, in the bundle sheath, remember the chloroplasts are composed mostly of stroma, and stroma is where that fixation is occurring for the Kelvin cycle. So therefore, Rubisco is only found in the bundle sheath cells when we're talking about C4 photosynthesis. In this figure, it compares and contrasts C3 versus C4 cycles. C4 plants have developed a way to suck in gas efficiently through partially closed stomata, delivering it later in high concentrations where the Kelvin cycle is occurring. Now in phase 1, carbon dioxide is drawn in the leaf where it is incorporated, concentrated, and stored in 1 or more compounds, all of which have 4 carbon atoms, hence the name 4C when we're talking about C4 photosynthesis. And in phase 2 of C4 photosynthesis, the carbon dioxide is released from storage in these compounds, and it is delivered to where it is needed. And this place that it is needed uses the same pathway that is found in all plants, the same as we see in C3 photosynthesis. So what are the effects of light and temperature on photosynthesis? Well, in the first graph on the left, we can see we have an increase in photosynthesis on one axis and an increase in light intensity on the other. And notice that as light intensifies, C3 plants level out before C4. Therefore, this tells us that C4 is able to do photosynthesis more efficiently at higher light intensity. The other graph shows us a quantum yield of photosynthesis, which is to say sugars produced, versus an increase in temperature. And in this case, as temperature increases, notice that C3 plants start to plummet. They're going to produce much less sugars at high temperatures because the stomata are closed. But C4 plants, because they have this special pathway that minimizes photorespiration, they're able to keep making sugars. All right, and our last type of photosynthesis is called CAM photosynthesis. And CAM is an abbreviation for crassulacean acid metabolism. CAM photosynthesis is an extreme adaptation to both heat and drought. It's been discovered in at least 20 plant families, but it gets its name from the stone crop family, which is the Crassulaceae. But we also see CAM photosynthesis in the cactus family, which is the Cactaceae, the orchids, and the bromeliads as well. CAM plants minimize photorespiration and save water by separating these steps in time between night and day. So this is kind of like a temporal relationship. Now at nighttime, CAM plants will open their stomata, allowing carbon dioxide to diffuse into the leaves. By opening at nighttime only, it is going to help the plants minimize water loss during the hot daytime. This carbon dioxide is fixed into an organic compound by PEP carboxylase enzyme at nighttime. And this is the same step that's used by C4 plants, with the difference being the temporal relationship. The organic acid is stored inside vacuoles until the next day. Then in the daylight hours, CAM plants close their stomata to minimize water loss, but they can still do photosynthesis because they have carbon dioxide. They stored it overnight by using PEP carboxylase to fix that carbon dioxide into 4 carbon organic compounds. Because these organic acids can be transported out of the vacuole and broken down to release carbon dioxide, which then enters the Kelvin cycle, this allows CAM plants to do photosynthesis during the day with their stomata closed. All right, so in review, we've talked about C3 photosynthesis, which is the normal photosynthesis, and we've compared and contrasted it to C4 photosynthesis and CAM photosynthesis. I hope that answers all of your questions you might have about photosynthesis, and please message me with any questions. Thank you so much for listening.